Pembrolizumab is an interesting drug because it was the first one ever to be approved based on genetic biomarkers, MMR deficiencies. And that's what this question's about. And it's very relevant to step one. So very exciting. 58-year-old man, history of colorectal cancer, and he's started on this medication, and it reveals a defective DNA mismatch repair gene. Compared to most tumors with intact MMR, why does this one respond better to this immunotherapy? Okay, so let's talk about this for a second here. Lynch syndrome, you've probably heard of, very important pass level for STEP. And Lynch syndrome, a lot of mutations occur, and these mutations will impact certain enzymes, like MLH1, MSH2, MSH6, and PMS2. Those enzymes are mutated in Lynch syndrome. And so these enzymes no longer work, and so DNA errors will accumulate, and you have microsatellite instability, MSI-H and MSI-high. And so the MMR system is responsible for correcting those errors, right? And if you have mismatched nucleotides, then these can be caught by the T cells. And so there's this like war going on with the tumor, and the tumor wants to use its whatever mechanisms it has in its power to make sure that the T cells don't work. And so it does this thing with PDL1, programmed death ligand 1. Tumors increase the expression of PDL1 to bind to PD1 on T cells, and that inactivates them. So PDL1 to the PD1 on T cells, now the T cells are inactivated. And now CTLA4 is cytotoxic T lymphocyte associated protein 4, outcompetes CD28 for B7 molecules on antigen presenting cells, and that will lead to decreased T cell activation. So there are ways that tumors can evade the T cells, and that's through the PDL1s. And so one thing we need to know about these weird tumors with MMR deficiency is that they produce more neoantigens. These neoantigens can be detected by T cells, and this increased neoantigen production is what makes them easier to kill. And so that's the reason why our answer is going to be E. Why is not A? Decreased regulatory T cell activity. Regulatory T cells suppress the immune system, but MMR deficient proteins are not linked at all to Tregs. So they just don't link to them. Downregulation of class 1 MHC expression. So some cancers do that. They reduce the expression of MHC1 so that they can hide from the cytotoxic aid cells, the T cells, but that's not how this one works. Increased production of metalloproteinases. Metalloproteinases help the tumor spread, but it wouldn't change how responsive you are to the therapies. Overexpression of tumor angiogenesis factors. So more blood supply to the tumor would definitely help it grow, but it wouldn't affect how it's detected by the T cells. So that's going to be our correct answer. So MMR deficiencies, more DNA mutations, more abnormal proteins called neoantigens, and then the check inhibitors help T cells recognize and attack those cancers better. And so that's the reason why this medication is going to work. So yeah, it's a pretty cool drug, pretty cool medication. Basically the checkpoint inhibitors blocking those PDL and PD1 interactions and blocking the CTLA4, the drugs restore the immune system's ability. So it's pretty freaking cool how it all works and is used in colorectal cancer, endometrial cancer, gastric cancer, and other cancers. And yeah, monoclonal antibodies, pretty neat stuff. That's all we got. If you liked it, like and subscribe.